So now we're going to talk about DNA replication in a little more detail. Last time we talked about just sort of the overview of how DNA replication works, some of the major principles. Now we're really going to talk about DNA polymerase, which is the enzyme responsible for most of the production of new DNA during DNA replication. So first of all, there are basically two kinds of enzymes that are associated with DNA. A polymerase is an enzyme that polymerizes DNA. So DNA, of course, is a polymer, and that it's a set of subunits that are connected together in a repeating chain. So polymerase is just an enzyme that makes a polymer. So DNA polymerases make DNA, RNA polymerases make RNA. All DNA polymerases work the same way. Like we said last time, they add nucleotides to the 3' end. So DNA polymerases always make new DNA from the 5' to 3' end, or we say that they write down. And they all, generally, at least the ones necessary for replication, require a template. The template would be the parental DNA. So usually with DNA polymerase, you have both strands of DNA acting as a template to one of the new daughter strands. As we'll learn later, RNA polymerase only requires one of the strands as a template. And then a nuclease is the opposite. It's an enzyme that breaks down nucleic acids. So DNA polymerase breaks down DNA, RNA polymerase breaks down RNA. RNA nuclease breaks down RNA. So nucleases can be categorized based on where they degrade from. So an exonuclease is a DNA molecule. So an exonuclease is an enzyme that breaks down DNA or RNA starting at either the 5' end or the 3' end and working in. So let's say this is our DNA, and let's say that's the 3' end of that strand, 5', 3'. So an exonuclease would start at one end let's say that it's the 3' end, and start breaking off the individual nucleotides. So you would have the enzyme, again, breaking up DNA and usually breaking off one nucleotide at a time, although some can break off larger chunks. And of course, assuming this is both ends are accessible by the enzyme, both ends will be degraded. So this enzyme would do the same thing from the opposite end. So this one going this direction, this one going this direction. So both of them presumably would just keep going until they got to the end, and then that piece of DNA would be gone. So this would be a 3' endonuclease because it starts at the 3' end of the DNA. There's the broken off pieces. And of course a 5' exonuclease would do the opposite. So again, this would be an exonuclease. Meanwhile, an endonuclease would start somewhere inside the molecules. So again, if this is your DNA, again, say this is 3', that's 5', then the enzyme in that case would start somewhere in the middle and work its way down. Make my lines. So in this case, you would then see eventually, once the enzyme started, you would see it starting to break down the DNA on both strands. They don't always work on both strands, but some do. So again, they're going to work this way. And same thing, they're going to be breaking off one nucleotide at a time, sort of chewing it up from the inside out. And then if this is linear, then they'll keep going until they get to the end. The difference between exonucleases and endonucleases, other than the way they start. So again, this is an endonuclease. So these guys start from the middle, and they always start from a particular recognition site. So most endonucleases have a particular sequence of bases that they look for to start cutting from. So these are the enzymes that are involved in DNA replication. Again, DNA polymerase is the enzyme that actually makes DNA, but nucleases are important too for degrading DNA, and they also have a role in repairing DNA, as we'll see in a second. So polymerases are the two kinds of enzymes that are involved in breaking down DNA and making DNA, respectively. But now we're going to focus mostly on DNA polymerase because we are talking about replication, and that is what DNA polymerase is largely responsible for. There are lots of kinds of DNA polymerases. This is just one. This is an example of a bacterial DNA polymerase, but all of them have at least the ability to bind to DNA, so they usually have this kind of configuration with a hand-like arrangement where the domains of the protein that bind DNA kind of wrap around it, and then an active site somewhere that allows the new nucleotides to be added. This is a more complex kind of DNA polymerase called DNA polymerase 3, and you can see it also happens to have 3 different polymerase core domains. So these are the parts that actually make new DNA, but there's 3 of them, and a number of components, including the sliding clamp and clamp loader, that are important for holding on to the template DNA and moving along the template DNA as the reaction proceeds. So if you look at the active site of a typical DNA polymerase, it will of course have space for the template DNA and the growing DNA strand to fit, and then space also for the incoming dNTP. So this is the new nucleotide that's going to be added to the 3' end. So in this case, you can see there's some components that are necessary for stabilizing those things. Those magnesium ions, for example, because they're positively charged, they help stabilize the 3' end, because the 3' end of the growing strand has can have a negative charge on it, and then the incoming dNTP, of course, has at least 2 or 3 negative charges on the phosphate groups. But when a new base is added, it comes into the part of the enzyme called the insertion site, and then it gets added, and then the enzyme simply moves down to the next base and allows the base that was just added to fill in what's called the post-insertion site. Then you can also see that at the active site, this is where the actual base pairing occurs. So in the active site, there's a spot for the new base that's about to be added and the base on the template strand that it should be complementary to. And the shape is such that only those Watson-Crick base pairs will fit stably inside that active site. So a CG or an AT base pair. Any other base pair, one of these over here, you can have base pairing between a T and a G, for example, but the active site of the enzyme will not allow that structure to fit stably. So that's how the enzyme essentially knows which base to add to the growing strand, because if it's not complementary, to the other strand, then it just won't fit in the active site. And then most DNA polymerases, or many DNA polymerases, also have an exonuclease ability, which is, of course, the opposite of a polymerase. But some of them can also break down DNA. So this is an example where the DNA polymerase has a 3' to 5' exonuclease activity, which is the opposite direction that it synthesizes, because this is its main, one of the first ways that a DNA polymerase can correct errors, because like I said before, the enzyme has this active site that should only fit these Watson-Crick base pairs, but there are other base pairs that could fit temporarily or less stably, and so sometimes the enzyme can just mess up and put the wrong base in. So in this case, for example, what it's done is it's added a C across from a T, which is not a good base pair. But what happens is when the enzyme does that, it forms this unstable structure because C and T don't base pair effectively, so the double helix shape no longer fits into the binding site of the enzyme, and so that makes it kind of change its conformation such that the insertion site is no longer aligned with the growing strand. Instead, this exonuclease domain or active site is present, and it gets close to now that base that's there. And so first thing it does is it just breaks off the last base that got added, which was the wrong base. And then now that base is, that position is empty, so the enzyme moves back into position, and the insertion site now is positioned over that T on the template strand, and the enzyme can wait for an A to show up to add into the growing strand. So that's one of the ways that the errors in DNA can be fixed. They can be fixed as essentially it's being made. And then there are lots of different kinds of polymerases. These are just the kinds of DNA polymerases in E. coli. If you were to look at eukaryote DNA polymerases, this will be a much longer list, but you can see there are different kinds that vary a lot in complexity. So DNA polymerase 1 and 4, for example, those just have one subunit, but if you look at DNA polymerase 3, it has all the way up to 9. They differ quite a bit in size. They also differ in their activity. So 1, 2, and 3, for example, in bacteria have the ability, have this exonuclease ability in the 3' to 5' direction, which allows for proofreading. But then DNA polymerase 1 also has a 5', 3' exonuclease ability. And that will become important later, but you may wonder what that is for, because of course 5' to 3' is also the polymerase direction. So why would it degrade DNA in the same direction that it makes it? And we'll talk about that later. They differ in polymerization rate, which just means the number of nucleotides they can add per second. DNA polymerase 3 is the fastest, which is good because it also has the highest processivity. That means the number of nucleotides that the polymerase will add before it just lets go. And so DNA polymerase 3 is the fastest and can add the most bases, and that's important because it is the enzyme that does most of the DNA replication in a typical bacterial cell. So next time we will get into the major steps of DNA replication.